Good afternoon, my dear friends. I'm Sol from Biological Sciences with Sol channel. Today I'm with new video of General Biology course, Unit 5. This unit is all about genetics and evolution. So today, in this particular video, I'm going to discuss protein synthesis. So, as usual, please don't forget to like and share this video and subscribe my channel. And let us proceed. Protein synthesis. So first, DNA, which is found in the nucleus, is transcribed into messenger RNA. Then messenger RNA moves from the nucleus to the cytoplasm and it is translated to a protein. So the transcription of DNA to messenger RNA and translation of messenger RNA to protein is the central dogma of molecular biology. And proteins can be source of food, and all the enzymes are protein, of course. These antibodies are proteins. There are hormones that are proteins, and there are proteins which are used as a transport mechanisms, or they are known as transport proteins. Our hairs and nails also made up of proteins. So all proteins that are found in our body are formed through this central dogma of molecular biology. That means DNA is transcribed into messenger RNA, then messenger RNA is translated into protein. Events during protein synthesis. First, transcription. This transcription is a process in which DNA codes for protein are written in a molecule of messenger RNA in the nucleus. DNA is a huge molecule that can't leave the nucleus, so it sends the genetic code through messenger RNA, and this written of DNA codes to a molecule of messenger RNA is known as transcription, and it occurs in the nucleus. Now, this messenger RNA travels from the nucleus to the ribosomes in the cytoplasm. Third, freely suspended amino acids in the cytoplasm are transported to the ribosome by transfer RNA. Now, ribosome, which is a site where protein is synthesized, it reads the messenger RNA codon and assemble amino acid which are presented by transfer RNA into a protein by a process known as translation. So translation is a process in which messenger RNA codons are converted into amino acid sequences to make protein. Steps in protein synthesis. The first step is transcription. So this transcription is the process by which genetic information in a strand of DNA is copied into new molecule of messenger RNA. Because DNA has two strands, so one of these strands is copied into new molecule of messenger RNA. And this transcription occurs in the nucleus of eukaryotic cells and in the nucleoids of prokaryotic cells. DNA is a double strand molecule, already we know that. And one of its strand is sense strand or coding strand, and the other strand is antisense or template strand. DNA is copied to messenger RNA through transcription using antisense or template strand as a template or as a mold. However, RNA, including messenger RNA, is single strand molecule. Here in this figure, you can see that DNA, which is a double strand molecule, the two strands of DNA are complementary to each other, meaning guanine bonded with cytosine, thymine with adenine, cytosine with guanine, adenine with thymine. In general, the two strands of DNA are complementary to each other. So this messenger RNA, which is transcribed from DNA, especially from template strand, is complementary to the template strand of DNA. Here, guanine with cytosine, uracil, because it is a messenger RNA, so instead of thymine, it is uracil. So uracil with adenine, guanine with cytosine, thymine with adenine. So the messenger RNA and the template strand of DNA are complementary to each other, but messenger RNA is identical to that of coding strand of DNA, except uracil in messenger RNA replace thymine in the coding strand of DNA. The second step of protein synthesis is translation. Translation is the process of translating the base sequences in messenger RNA. These base sequences in messenger RNA are also known as messenger RNA codons. So translation is the translating of this messenger RNA codon into amino acid sequences of protein molecule. It occurs in the cytoplasm, especially on the ribosomes, and further this step is subdivided into initiation, elongation, and termination steps. The first step of translation is initiation. So mostly protein synthesis is initiated or started by adenine, uracil, guanine, codon on messenger RNA. So once this codon is found, transfer RNA with anticodon, uracil, adenine cytosine holding amino acid methionine will start the process. Here in this figure, we can see that messenger RNA and on the first codon or start codon, adenine, uracil, guanine, this codon codes for methionine. So transfer RNA with anticodon, uracil, adenine, cytosine. These base sequences are complementary with adenine, uracil, guanine. This codon, which codes for methionine, brings the methionine. So the first amino acid on protein is methionine. So this is initiation step of translation. The second step of translation is elongation. Once protein synthesis is started by methionine, then the protein must be grown or elongated by bringing different amino acids. So the second codon on messenger RNA and the second transfer RNA molecules enter the ribosome, and the peptide bond is formed between carboxylic acid group of the first amino acid and the amino group of the next amino acid. Here in this figure, you can see that messenger RNA with different codons. Here, the first codon codes for methionine. So this methionine is initiating amino acid. Then the second transfer RNA brings threonine, which is coded by adenine, cytosine, guanine, and there is peptide bond between these two amino acids. And the third transfer RNA brings glutamate, which is coded by guanine, adenine, guanine, and there is peptide bond between threonine and glutamate. The fourth transfer RNA brings leucine, which is coded by cytosine, uracil, and there is peptide bond between glutamate and leucine. And still there is the fourth transfer RNA brings arginine, which is coded by cytosine, guanine, and there is a peptide bond between leucine and arginine. And the fifth transfer RNA, I think the sixth transfer RNA brings serine, which is coded by adenine, guanine, cytosine, and there is a peptide bond between arginine and serine. And finally, there is no amino acid, which is brought here because this codon codes for no amino acid. It is a stop codon. So at this point, protein synthesis is terminated. The third step of translation is termination. When a stop codon on messenger RNA is reached, the protein synthesis is terminated and the peptide chain leaves the ribosome. The ribosome is now free to repeat the process, and the three stop codons are uracil, adenine, uracil, adenine, guanine, and the uracil, guanine, adenine. These three codons are stop codons, so whenever the process reaches on this one of these stop codons, then protein synthesis is terminated. So this termination is the third step of translation. Here is to add some note. Translation of messenger RNA codons into protein molecule requires energy. This process, translation process, requires energy, and this energy is obtained by hydrolysis of guanosine triphosphate, that is GTP, not ATP. Amino acids are detached from their transfer RNA because once transfer RNA brings the required amino acid, then they are detached to each other. This is by hydrolyzation process. And the third, reactions of formation of peptide bond. Once amino acids are brought to the ribosomes, and there is a formation of peptide bond between the consecutive amino acids. So this peptide bond formation is catalyzed by an enzyme known as peptidal transferase. Already we know that translation of protein from messenger RNA is carried out on ribosomes, and each ribosome has 3 distinct sites. The first site is P site, or it is known as peptidal site, which carries growing peptide chain until protein synthesis is terminated and the peptide chain leaves the ribosome. And the second site is A site, or it is amino acyl site, which carries new incoming transfer RNA with the amino acid that is going to be added next. And the third site is E site, or exit site, is a site through which transfer RNA is get out of the ribosome by leaving its amino acids. For example, if this is ribosome and it has 3 parts or 3 sites, and the middle site is peptidal site, which carries growing peptide chain until protein synthesis is terminated and the peptide chain leaves the ribosome. And this one is E site, or exit site, through which transfer RNA gets out of the ribosome by leaving its amino acid. And this one is A site, or amino acyl site, which carries new incoming transfer RNA with the amino acid to be added next. So this can be entrance for transfer RNA. This can be exit for transfer RNA by leaving its amino acid, and this peptidal site can be, or it carries growing peptide chain until protein synthesis is terminated and the peptide chain leaves the ribosomes. The next point is about features of genetic codes. And the first feature of this genetic codes is they are triplet codons. Each codon is consecutive sequence of 3 bases on messenger RNA. For example, uracil, 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 which codes for phenylalanine, uracil, uracil, adenine, which codes for leucine, and HC. And the other feature is they are non-overlapping in nature. The codons are consecutive and non-overlapping. Therefore, the starting point is extremely important. Once the starting point is obtained, then it is to group these genetic codes into three groups. Therefore, for example, they are already consecutively adjusted. However, in our mind, we have to adjust three bases in one codon, three bases in one group. So they are what? They are non-overlapping in nature. The other feature of genetic codes is non-punctuated. They are non-punctuated. There is no punctuation between codons. There is no need for comma or any other punctuation to separate one codon from another. So they are found continuously or consecutively. And the other feature is they are universal. The codons are the same for the same amino acid in all the species. The same codon codes for the same amino acid in all the species. It codes for elephant and it codes the same for Escherichia coli. The same codon codes for amino acid for plant and for animal. Therefore, it is believed that genetic code has been highly preserved during evolution. The other feature is degeneracy nature of this genetic code. So they are degenerate. There are 61 codons that code for 20 amino acids. So one amino acid has more than one codon, but not one codon codes for more than one amino acid. For example, serine has 6 codons. Here in this figure you can see these 4 codons are here and the other 2 codons are here. Still, glycine has 4 codons here. Glycine is coded by 4 codons. So this nature of genetic codes is degeneracy of their nature. The other feature is terminator codon. They terminate the protein synthesis. There are 3 amino acids which do not code for any particular amino acid, so they are nonsense codons, or they are punctuator codons, or they are terminator codons, or stop codons. So they stop, full stop to protein synthesis. These stop codons include uracil, adenine, adenine, uracil, adenine, guanine, and uracil, guanine, adenine. Uracil, guanine, adenine is a stop codon, but in special case it codes for the selenio synthesis, cysteine, which is 21's amino acid. Finally, this genetic course can be initiator codon. In most cases, adenine, uracil, guanine acts as initiator codon, which codes for methionine. But in few protein, this guanine, uracil, guanine may be initiator codon that codes for valine. Thank you for watching, and I'll come back soon with another new video, and until that, see you. Bye bye.